Picture two 50-year-olds. According to their birth certificates, they have been alive for the exact same amount of time. But watch how they move. Underneath the surface, the differences are even more stark. One subject possesses the clean, elastic blood vessels and sharp cellular function of someone decades younger. The other has cells actively struggling to repair basic wear and tear. For centuries, medicine has relied almost entirely on the calendar to assess baseline health. A patient turns 50, and standard protocol tells doctors to start screening for heart disease, cognitive decline, and arthritis. The flaw in that system is that human biology does not own a calendar. Age-related diseases look for systemic cellular degradation, not the number of candles on a birthday cake. Researchers have long known that people age at wildly different rates. But checking every single organ and tissue simultaneously to calculate an accurate systemic biological age was technically impossible. To actually slow down or stop the physical aging process, we first needed a reliable way to measure it. We needed a biological clock. In 2013, researchers pinpointed exactly where the body keeps time. They discovered aging leaves a distinct chemical footprint directly on our DNA sequence. Think of your genome like a computer's hardware, the static code you're born with. The epigenome is the software. It dictates which lines of that code the body should actively read and which lines to ignore. Over time, the body accumulates tiny chemical tags known as DNA methylation. As these tags build up, they begin to silence crucial genetic instructions, effectively shutting down youthful cellular repair functions. A researcher named Steve Horvath realized these methylation patterns were highly predictable. He trained an artificial intelligence model to read these exact patterns across various tissues to perfectly estimate a subject's chronological age. This algorithm, known as the Horvath clock, proved definitively that a unified, trackable aging process runs simultaneously through the entire human body. But predicting a person's calendar age was only the first step. The true objective for longevity science is predicting when the machine will break. This requirement led to a second generation of epigenetic clocks. Models like GrimAge abandoned calendar age entirely, focusing strictly on assessing mortality risk. As this diagram shows, GrimAge calculates risk by analyzing methylation patterns explicitly connected to a history of smoking and specific proteins that signal organ failure. It measures how degraded the system actually is. Then came a third leap in the science, DunedinPACE. Instead of tallying total cellular damage over decades, it measures the exact velocity of aging right now. If GrimAge is the odometer tracking miles driven, DunedinPACE is the speedometer, tracking whether a person ages faster or slower than one biological year per calendar year. Possessing a real-time biological speedometer takes longevity research out of the realm of long-term guesswork and turns it into an exact, trackable science. Before these tools existed, testing any anti-aging intervention required an agonizing wait. Researchers had to follow human subjects for decades until they eventually died just to prove a drug actually extended lifespan. Epigenetic clocks bypass that bottleneck entirely. By reading the methylation patterns before and after an intervention, scientists receive highly accurate, measurable longevity data in a matter of months. Programs like the National Institute on Aging's Interventions Testing Program rely on these exact biomarkers to scientifically validate pharmaceutical compounds that might extend healthy life. They run a rigorous testing protocol across independent laboratories, utilizing genetically diverse mouse populations to ensure the results are highly reproducible and not just a statistical fluke. This measurement capability extends far beyond pharmaceutical trials. Everyday lifestyle choices, how much you sleep, what you eat, and how you manage stress actively alter your methylation patterns. Your chronological age marches forward inevitably, but your biological age remains highly flexible, responding directly to the daily interventions you apply. The epigenetic clock acts as a dashboard. For the first time in human history, we have the metrics required to steer our own aging.